This is the Soul Cycle. In our group, there's me, Anthony Diaz, and then my partners, Leon Perez and Jose Rodriguez. To start off, cell division is needed for the development from a fertilized cell, for the growth and the repair of the cell. Two types of cells are the gametes, which are the sex and sperm cells, and then every other cell are called somatic cells. All the DNA in one cell is called a genome. For the basics, here you have a chromatin, which is DNA and the protein, and this protein, they're called histone proteins. Here you have a duplicated chromosome, which is referred to as sister chromatids. And here you have a centromere in the middle, and the telomeres will be every each of the ends. On just a chromosome is an unduplicated one, and then here homologous chromosomes are just two duplicated chromosomes, or sister chromatids. The stages of the cell cycle, it starts off in a G1 phase. Here the cell grows and carries out the normal cell functions. Then you go to the S phase, which is synthesis, which is synthesizing the DNA. The chromosomes are copied, developing the cell's DNA. Then you go on the G2 phase, which is the cells. It continues to grow and produce chemicals that it needs to divide. And then when it divides, it goes into, it's called the mitotic phase. It starts with meiosis, which is prophase, prometaphase, metaphase, anaphase, and telophase, which we will learn more about in the future slides. And then the separation of the cytoplasm is called cytokinesis.
Okay. There are different checkpoints in the cell cycle along the different phases, such as G1, G2, and S phase. And now there's a specific checkpoint at G1, which if the cell does not receive the signal to go, it enters a phase called G0, which it will exit the cell cycle and the cells will no longer divide, it will no longer grow, and the DNA will not be replicated. If it does receive the go-ahead signal, it will go through the rest of the cell cycle as planned.
Next in the cell cycle, we have prophase. Prophase is basically each duplicated chromosome becomes two identical sister chromatids joined at their centromere, and it also has nucleoli, which disappears basically in that phase. Next, we have the prometaphase, which some of the microtubules attach to the kinetochores, becoming kinetochore microtubules, which are responsible for moving chromosomes. Nuclear envelope, we also have that in the prometaphase. We have nuclear envelope fragments, and each of the chromatids now has a kinetochore protein at the centromere. In metaphase, we have centrosomes, which are opposite ends of the cell. Chromosomes are lined up at metaphase plate. We also have the anaphase, which the two daughter chromosomes begin moving towards opposite ends of the cell, separates the chromosome into daughter chromosomes, and the cell elongates. Last but not least, we have the telophase. Telophase is basically two daughter nuclei form, and nuclear envelope begins to form. The nucleoli reappears back, which we already went over that it disappeared earlier. Reappears back. Also, the spindle fibers are depolymerized. Last but not least, we have the cytokinesis, which is the division of the cytoplasm is usually well underway by late telophase. Therefore, the two daughter cells appear shortly after mitosis ends. In animal cells, cytokinesis occurs by a process known as cleavage, forming a cleavage furrow. And lastly, in plant cells, a cell plate forms during cytokinesis.
That will be all.